Ralph Baric is often referred to as the godfather of gain-of-function experiments. His lab at the University of North Carolina has pioneered groundbreaking techniques in virus manipulation and reconstruction. Those techniques were used on bat viruses at the Wuhan Institute of Virology, with whom Baric has cooperated since at least 2015. More recently, Baric has downplayed his association with the Wuhan lab, going so far as to suggest that the lab's notably lax safety standards may have caused the pandemic. Baric's statements have set him on a collision course with the Chinese Communist Party, who denounced him in two recent statements. We look at the history between Baric and the Wuhan lab and assess the CCP's new fixation on Baric. Hi everyone, and welcome to Truth Over News with Jeff Carlson and Hans Malmke.
In late August 2021, Chinese embassies in Washington, D.C. and in Paris openly insinuated that COVID-19 may have been created in a lab, and they highlighted the work of University of North Carolina professor Ralph Baric. The Chinese embassy in Paris tweeted that in December 2019, before the outbreak of COVID-19, Peter Daszak had admitted that his alliance had cooperated with Baric to modify coronaviruses. The Chinese embassy in Washington also directed their attention towards Baric when they issued a statement noting that Baric possessed extremely mature capability in synthesizing and modifying coronaviruses. The timing of these messages from the Chinese embassies is notable, as they came in front of a leaked 2018 proposal from Peter Daszak that included Baric's lab. The messages may also have been a preemptive effort to deflect blame from China, as Daszak's proposal included the Wuhan Institute of Virology and his longtime collaborator Shi Zhengli. Daszak's proposal laid out plans to create entirely new coronaviruses through the synthetic combination of pre-existing virus backbones. It also described how those viruses were going to be made more virulent in humans by the insertion of a furin cleavage site, a feature that distinguishes COVID-19 from all other SARS-related coronaviruses.
The Chinese embassies were not the only ones to point their fingers at Baric. Lin-Fa Wang, the Wuhan lab-connected scientist who confirmed the authenticity of Daszak's proposal, was recently asked who had first proposed to insert furin cleavage sites into bat coronaviruses. Wang initially claimed that he didn't know. But when the question was repeated, Wang blamed Baric, saying UNC, University of North Carolina.
UNC, right? It's a UNC lab. The grant is very clearly.
It's University of North Carolina. In case
Yeah.
In some respects, the Chinese embassies were correct in pointing out Baric's role in pioneering the creation of synthetic viruses. Baric, who is known as the godfather of gain-of-function research, pioneered reverse genetics techniques that now allow scientists to engineer viruses with altered functions. As early as 2008, Baric computed a consensus sequence of a bat coronavirus that was an amalgamation of 4 existing bat coronaviruses. At the time, it was not possible to reverse engineer the consensus sequence into a live virus. But Baric's new technique now makes it possible to construct viruses from their genomic sequence. In November of 2015, Baric and Shi Zhengli, the director of the Wuhan Institute of Virology, famously co-authored an article in the science journal Nature. That article appeared to showcase Baric's reverse genetics techniques, claiming they had used the reverse genetics system to generate chimeric viruses that expressed the spike of a bat virus in a mouse-adapted coronavirus backbone. It was this article that set off warnings about the dangers of gain-of-function experiments. prompting Simon Wayne Hobson, a virologist at the Pasteur Institute in Paris, to warn that researchers had created a novel virus that grows remarkably well in human cells, and that nobody could predict the trajectory if the virus were to escape.
Baric has repeatedly defended his life's work, saying such research is essential to the development of vaccines and other countermeasures against emerging viruses. Baric has also disputed Xi's level of participation in his November 2015 paper. He has stated that he first met Xi, the director of the Wuhan lab, in 2012 or 2013, when she was presenting her work on 2 new viruses that Xi had discovered. Baric said he requested the sequences of those viruses, which Xi supplied to him, and he claimed that was the basis of that collaboration. Baric and his team then took that sequence and inserted it into a genetic copy of the SARS virus that Baric had in his lab. When the modified virus was introduced to mice and human airway cells, it exhibited robust replication. Baric went to some lengths to distance himself from Xi, claiming in an interview that we never provided the chimeric virus sequences, clones, or viruses to researchers at the WIV, and Dr. Xi or members of her research team never worked in our laboratory at UNC. Baric also said that no one from his laboratory has ever worked at Xi's institute. But we do know that Baric provided the transgenic mice with human lung receptors to Wuhan under a material transfer agreement. And we also know that Baric and Xi have co-authored at least 3 scientific articles beginning in August of 2015. What is not in dispute is that the Wuhan lab quickly gained access to Baric's technology, as illustrated in a June 2016 article from Shi Zhengli, in which she described gain-of-function experiments carried out by her lab. Also, and perhaps more ominously, a 2017 doctoral thesis detailed how Zhengli was already performing experiments that replaced the S gene without traces on bat coronaviruses. In other words, she was using seamless technology whereby no evidence of lab manipulation would be visible. Baric has since confirmed that it was possible for the Wuhan Institute to manipulate viruses without leaving traces, admitting that you can do it without leaving a signature. Using any of 3 or 4 different approaches for coronaviruses that were developed by different researchers, you can leave no trace that it was made in a laboratory. Baric was a pioneer in this area. His reference to 3 or 4 different researchers may have been an attempt to deflect attention from his own collaboration with the Wuhan lab. It may also reflect a genuinely held belief that Shi Zhengli did not acquire the technique from Baric's lab and had actually learned it elsewhere.